Dr. Alonso leads diabetes research lab at Weill Cornell Medicine, studying the basic biology of pancreatic beta cell regeneration. She serves on multiple journal editorial boards, co-chairs an NIH grant review panel, and is on the organizing committee for the scientific sessions of the American Diabetes Association. In 2019, she was elected to the American Society for Clinical Investigation. She mentors trainees at all career levels, and she also, in addition to teaching and research, also sees patients at the 80th Street Cornell Medicine Endocrinology office. And today she has a talk for us to grow new pancreatic beta cells, putting the old ones to work. Thank you so much.
Thank you very much for that introduction. Thanks for the invitation to be here. I'm going to add a couple of things to what you did. That was a really kind introduction. I do want to mention that my first faculty position was in University of Pittsburgh with working for Andy Stewart, one of your own. I don't want to leave that out. And yeah, and I would like to say that I do really hope that I get to meet all of you guys at some point for real, rather than in this format, which is, you know, really convenient, but not so good for actually getting to know each other. So I will start. Whoops, if I can advance my slides. So I do have a scientific disclosure, which is not relevant to this current presentation and may not be relevant at all anymore. Okay, so what I'm going to talk about today is several things. First, because as you may know, we are now in the 100th year of therapeutic insulin. I'm going to talk briefly about the history of the discovery of insulin, which is pretty inspirational. And I personally like to take a moment to reflect on how far we've come. And then I will talk about, you know, where do new beta cells come from in the adult pancreas, with the idea that, you know, insulin deficiency is a problem and beta cell regeneration is an important potential solution to that problem. And then I'll share with you some of my own work from my research lab, which started in Pittsburgh, as I mentioned, on beta cell workload and beta cell stress and how that leads to regulation of pancreatic beta cell number. And I'll touch briefly on implications for the future. What you see on the right here are this is mouse pancreatic beta cells stained for insulin in green. You can almost, I don't know if these are actual insulin granules, but you can see that there seems to be more insulin lined up around the edge of these beta cells as if they're ready to secrete. And then blue is DAPI, which is a nuclear marker. So first, let's talk about insulin. So as you may or may not know, insulin was discovered in 1921, 100 years ago. And you definitely all should know that insulin exists in a hexamer. So this is an artist's rendition of the insulin hexamer that is very beautiful. So way back in ancient Greece, diabetes had already been recognized and described. And this almost certainly reflects insulin deficient diabetes, type 1 diabetes. And before the advent of therapeutic insulin, the life expectancy for someone with established type 1 diabetes was less than a year. Diabetes mellitus refers to the Greek for sweet siphon. And Areteus described diabetes as a meltdown of the flesh and limbs into urine. Its course is of a cold nature. The patients never stop making water as if from the opening of aqueducts. Did I say Greece? Well, he was Greek, but some of this is, now I'm confusing myself. This sounds Roman. The nature of the disease is chronic and it takes a long period to form, but if the constitution of the disease be completely established, the melting is rapid, the death speedy. So before insulin was available, we had MNT, not really as effective as today. The Allen diet consisted of thrice boiled vegetables, which, as you might imagine, might have prolonged life, but probably was not pleasant and was not terribly effective at treating type 1 diabetes. So this is a picture of Frederick Banting. He was a 28-year-old GP in Canada. He went to medical school at the University of Toronto, which was cut short due to World War I, and he actually served in the Canadian Army in France. And then he returned in 1919 to serve as a general practitioner in London, Ontario. And as the story goes, he was preparing for a lecture on pancreatic anatomy. And he read the following: any reference to the pancreas as secreting a hormone necessary for the utilization of sugar by the tissues of the body is misleading, as that function is exercised by only a very small portion of the organ, the so-called islets of Langerhans. So they knew that the islets were regulating sugar levels. So reportedly in the middle of the night on October 31st, Halloween of 1920, diabetes, ligate pancreatic ducts of dog, keep dogs alive till acini degenerate, leaving islets. Try to isolate the internal secretion of these to relieve glycosuria. So he basically wanted to cause pancreatitis, kill off the exocrine pancreas, and then try to scrape the islets, the remaining tissue from the pancreatic bed. So he pitched this idea to John MacLeod, who was head of physiology at the University of Toronto, who invited Banting to come and perform the experiment in the spring of 1921. So the experiment that they did was as he had described. So first, they would do a total pancreatectomy in a dog to make a diabetic recipient dog. And then in the donor dog, they ligated the pancreatic duct until the exocrine pancreas degenerated. And then they would prepare a pancreatic extract and then inject that pancreatic extract into the pancreatectomized diabetic dog. And after several failures, they actually had a diabetic dog pictured here who had been in a diabetic coma. And after injection of this pancreatic extract, the blood sugar dropped and the dog woke up from the coma in order to take this photograph. And I should mention that he was assisted by Charles Best, who was a fourth-year medical student at the time. So IRB, I believe, was probably not fully established at that time, but so that was this was August of 1921. And then already by December of 1921, they were injecting this into people. So Leonard Thompson was, I believe, the first recipient of the pancreatic extract. He was born in 1908 and he was diagnosed with diabetes in 1919. He was admitted to the hospital in December of 1921. At the age of 13, he was only 65 pounds and he was in ketoacidosis. And this is reportedly from the medical record that he was in December 4th, he was feeling well since admission, drinking fluids freely. December 31st, weight 65 pounds, diacetic acid still persists. January 11th, 15 cc's of MacLeod's serum, 7.5 cc's into each buttock was injected. That's the pancreatic extract. And then a week later, they noted an area of induration, circular 7.5 centimeters in diameter over the left buttock, which they had to incise and drain. But he survived until 1935, which is a really dramatic departure from the expected survival. And I'll highlight just one more patient back then. So this is Elizabeth Hughes and her family. I believe that this is Elizabeth. In her case, the diabetes was diagnosed in 1919 at age 11 when she was 75 pounds. She went on that Allen treatment, nutritional treatment, until at age 15, she was only 45 pounds. In August of 1922, insulin began, which she described as simply too wonderful for words. She regained weight. So by January of 1923, she was 105 pounds. And she ended up going to Barnard College, had 3 children, and died at the age of 74, which is completely amazing. So we should all be very grateful for insulin. So that was already amazing, right? However, we have certainly come a long way since then. technologies are improving very rapidly. An important game changer was the cloning of the human insulin gene. Until that time, only animal insulins were available for therapeutic use. If anyone is interested in the history, this book that David Harlan lent me is really a sort of, if you are into scientific thrillers and intrigue, this is a real true story of the competing teams that were working to try to clone the human insulin gene. As you're all well aware, we now have modifications to the insulin molecule that are engineered to change the duration of action of insulin. We can administer insulin through different routes of administration. There are on the horizon smart insulins that I'll talk about briefly in a slide or two. And of course, the advances in glucose monitoring and insulin pumps and closing the loop have really dramatically changed our ability to treat people with type 1 diabetes. And I won't call this out later, but I think every one of us knows how important it is to have a team of people in order to effectively treat, replace insulin in someone with type 1 diabetes. So just a brief word on the insulin analogs. What you all should know is that, so here's a diagram of pre-proinsulin with the A chain, the B chain, and then the C peptide. And then proinsulin after cleavage of the peptide. And then after cleavage out of the C peptide, you end up with mature human insulin, which is the A chain and the B chain that are disulfide linked. And most of the modifications that affect the onset of action of insulin are due to amino acid substitutions in the B chain. And that's to make shorter acting, including, lispro, aspart, and glulisine, which we use all the time. Fiasp is a new, as you know, new agent that actually is basically a new formulation, just with arginine added as a stabilizer. And then the long acting, there are several different approaches to allow insulin to be very long acting, including also some amino acid substitutions and in some cases, fatty acid moieties that are added to the molecule in order to extend half-life. And I'm sure that you also all saw that we now have once weekly insulin icodec, which was reported in the New England Journal back in September, with a half-life of 196 hours due to an albumin-bound circulating depot, due again to a fatty acid that's been added. So you are all well aware of these, but continuous glucose monitoring has been an absolute game changer, as have the various pump and pen smart insulin delivery devices. And honestly, if you sort of think about where we started so many years ago, the tools we have now are literally science fiction. You know, we have time in range, average glucose over a 24-hour period, what percent of the time the patient may be hypoglycemic, the glycemic variability. You know, we have tools now that we really couldn't have been, I think, conceived back then. You know, patients come in and we have honestly almost too much information in terms of how meals, exercise, and sleep affect their blood sugar, and life-saving information on, alarms, sharing, trend arrows. And then I'm not going to get into this, but the closed loop systems are already, I think, dramatically improving our ability to deliver insulin and are only getting better. So on the horizon, Afrezza is not on the horizon, it's here. There are under development insulin patches that will be able to deliver insulin directly through the skin. This is an example of a hypothetical smart insulin in which the insulin is encoded in a polymer that if the glucose is high, releases insulin, such that insulin would only be active in theory when it's needed during hyperglycemia. And there's a lot of technology on the horizon, especially with respect to glucose sensing, including smartwatches, wearable sensors, contact lenses, implantable sensors actually already, we already have one of those, patch sensors, non-invasive finger stick, sweat-based sensors, and probably more. So really, I think we should take a moment to celebrate just how far we have come. However, as you all are very much aware of, we have not solved this problem, the greater problem of diabetes. So, from the NHANES survey, this is, now a few years old, but only 50% of US adults with diagnosed diabetes had an A1C in what we would probably mostly consider to be the target range for most patients. And even though that is a few years old, I don't think the numbers are any better today than they were then. So now let's sort of step back a little bit and talk about the greater diabetes, syndromes. So insulin deficiency is a feature of all forms of diabetes, but the lean type 1 individuals that we've been talking about, from 1920 are, they just have a lower insulin requirement than overweight or obese individuals with type 1 or type 2 or gestational or any of the other, diabetes syndromes. But insulin deficiency is a requirement for hyperglycemia relative, insulin deficiency relative to body need. And so I think that the most effective cure, if we're going to use the word cure, would be restoration of normal endogenous insulin production. So this is really the focus of my lab. As you all know, insulin and glucagon are made in the pancreatic islets of Langerhans. So the islets are, as they knew way back in the early 1900s, in the islets of Langerhans, which are embedded in the exocrine pancreas. Human islets, this is from the Encyclopedia Britannica, are a mixture of alpha, beta, delta, and some other cell types that are distributed throughout. And of course, there's vascularization, innervation as well. This is a picture of a mouse islet from my lab, illustrating just the difference in architecture between mouse and human islets. Green are beta cells, and red is alpha cells, stained for glucagon. So the long-term goals of my lab are to understand the basic biology of pancreatic islet cell self-renewal and tissue homeostasis in order to ideally one day restore insulin production under normal physiologic regulation by regenerating endogenous beta cells. And this is a picture of dispersed mouse islet cells in culture, stained for insulin in green, nuclei, DNA in blue, and then these pink nuclei are beta cell nuclei, because they also stain for insulin, that also incorporated BRDU, which is bromodioxyuridine, which is it's an analog of the thymidine nucleotide that incorporates into cells during S phase. So these insulin positive mouse beta cells that have this pink nucleus are cells that actually have entered the cell cycle during this culture period. And so this is a model that we're using to try to identify what can trigger beta cells to enter the cell cycle. So Alice mentioned that I did part of my fellowship at NYU, and no, I didn't actually flunk out of my first fellowship. I was 2 years into my fellowship at the University of Chicago, working with Elaine Fuchs, pictured here, when she moved her lab from Chicago to Rockefeller. And so I had started already my research project, but I wasn't yet board eligible because I'd only done 2 years out of my ABIM research pathway 4-year fellowship. And so I somehow managed to get NYU to agree to help me continue my training there. And that's why I ended up with my fellowships, both from University of Chicago and from NYU. So in the Fuchs lab, what she was very laser focused on, and she studies skin, is stem cells. And although people knew that there were stem cells, they had really not been molecularly characterized up until that point. So this diagram over here is a hair follicle with the hair shaft coming out the middle, the dermal papilla, which is a dermal component, but the rest is epithelial, the outer root sheath and inner root sheath. And of course, you know, the hair shaft is, this is a very rapidly proliferative organ creating tissue that, you know, that exits the body in the form of hair. And all of the cells that lead to the layers of the hair shaft and the hair follicle, as well as the sebaceous gland and the interfollicular epidermis, the stem cells reside in this compartment called the bulge. And even the melanocyte stem cells are actually in the bulge as well. So in the Fuchs lab, I was studying what's called transit amplifying cells, which are the after the stem cell divides, it releases a cell that is sort of still a little bit stem-like, but proliferating rapidly and then and differentiating. And at that time, I think in our lab, certainly, and also really in general, I think everyone was very caught up in the idea of stem cells, and there was a general concept that all throughout the body, all of the tissues must have a stem cell compartment. So after I finished in the Fuchs lab and got my first position with Andy at University of Pittsburgh, it was kind of a big shock to me based on my training that there really was not a stem cell compartment in pancreatic islets. I think this is still a little controversial, but there's pretty good evidence, I think, that there is not a stem cell compartment that leads to new beta cell generation in adult mice. This is Doug Melton and Yuval Dor, and they published a really seminal paper back in 2004 showing that new beta cells come from pre-existing adult beta cells. And there are now, I think, many studies that support this observation. So what then are the determinants of beta cell mass? First, developmental accrual. So of course, if you're born with more, then you may have more as an adult. And there's, by the way, a really wide variation in human pancreatic beta cell mass, like up to 10-fold among normal adult humans without diabetes, according to Dave Bloom. Generating more beta cells can happen through proliferation of mature beta cells or generation of beta cells from a different cell type, termed neogenesis or transdifferentiation. In theory, you can increase beta cell mass by beta cell hypertrophy, meaning making each cell larger. I don't think there's really good evidence for this, that this happens on any major scale in the adult, in the pancreas at all, really. And then, of course, if your goal is to increase the number of beta cells, then you had better prevent them from dying or from de-differentiating. So this is over the past 10 years, I would say, has become a major focus of the islet field because we now recognize that beta cells can maybe persist in the pancreas, but lose some of their characteristics and therefore lose the ability to produce insulin and secrete it in response to glucose, which is functionally really devastating for insulin production. So it's been known for a really long time that mouse pancreas somehow has the ability to generate new beta cells. So if you take a normal, healthy, cute little mouse and you increase its insulin demand, and there's several ways to do that. So you can make them genetically insulin resistant through, knocking out insulin responsive genes, for example, insulin signaling genes. You can raise the blood sugar, can make them obese through diet or genetics. During pregnancy, that also increases insulin demand. Or if you increase insulin demand on a per cell level through either partial pancreatectomy, so you remove some of the beta cells and then the remaining beta cells have to work harder, or streptozotocin, which is a drug that kills off beta cells. If you partially kill off the beta cells, the remaining beta cells have to work a lot harder. So in all of those cases where there's unmet insulin demand, we think of this as increasing beta cell workload, and then something magical happens and beta cell number increases, restoring or maintaining glucose homeostasis. So the main two questions that my lab has been focused on are, how does the mouse pancreas generate new beta cells in response to demand? And the human pancreas seems to have limited capacity to expand beta cell mass. So why is the regenerative capacity blunted or lost? Although Dr. Stewart has made great strides in making human beta cells replicate. So way back when I was, a postdoc on the T32 in Pittsburgh, I devised the following experiment with my colleagues, principally Chris O'Donnell, who is a pulmonary physiologist in whose lab these experiments were performed. So we took mice, and these were based on, there were a series of studies done in rats that showed that raising the blood sugar through intravenous infusion was a stimulus for beta cell replication in the live rat. And rats at that point were really not genetically nearly as useful as mice. And so my idea was to translate that hyperglycemia model from rats to mice so that we could then study the genetics of the beta cell expansion response. So Chris's surgeon put in tiny femoral artery and vein catheters in the little mice. And we did a 4-day continuous IV infusion of either saline as a control or glucose, and then took the pancreas for histology or isolated islets to do molecular studies. During the 4-day infusion, the blood sugar was highly significantly increased, although quantitatively really not that dramatically increased. And the insulin levels in circulation increased quite a bit though. So clearly the islets, the beta cells, were seeing this short, small delta in blood sugar. And at the end of the experiment, if we took the pancreas and cut sections, you could find islets that are stained for insulin. Under the saline control conditions, there really wasn't a lot of replication marked by BRDU, but in the hyperglycemic mice, you could readily find proliferating, beta cells that had been through the cell cycle. And the quantification of that showed a dose-dependent increase in beta cell replication with infused glucose. Andy, from the very beginning, you should know, was very focused on human beta cells. And does any of this apply to human? Because as he loved to say, we really don't care about curing diabetes in mice. And so at his behest, we did this complicated experiment, which took non-skid mice. So these are immunodeficient mice. We killed off the mouse pancreas using streptozotocin. We engrafted human islets, gave the grafts 2 weeks to revascularize, and then we did our 4-day infusion of saline or glucose, again with the replication marker infused IV for the whole 4 days of this of the infusion. And then we were able to sacrifice the mouse and find the graft and actually identify human beta cells. And occasionally we were able to find human beta cells that appeared to have entered the cell cycle as determined by co-labeling with BRDU. This is showing you there were 4 donors. So we tried to take islets from the same human donor and put them in different mice so that we could have study the same islets exposed to different glucose levels in vivo. And for all 4 of the donors for which we achieved that, what you can see is the replication percent was higher in the beta cells that were exposed to the higher blood glucose level in vivo. So suggesting qualitatively that human beta cells exposed to a higher circulating blood sugar had a higher rate of beta cell replication. So this is the graph I showed you before with the mouse beta cells. And as you'll notice, the axis here goes to 6% of the beta cells labeled, well, about 4.5% labeled over the 4-day BRD labeling period. And in the human islet grafts, about 0.6% were able to label. So the human beta cell cycle entry with mild hyperglycemia was about 10-fold lower frequency than mouse beta cells in the endogenous pancreas with the same hyperglycemia. So again, this is now I'm going to share with you the main focus of what my lab studies now and going forward, which actually started all the way back in Pittsburgh, so we knew that raising the blood sugar can trigger beta cells to replicate, but we didn't know molecularly at all what was causing that. And we did test many different hypotheses and learned a few things along the way, but I wanted to find something new, something exciting. And so we did what's called a screen. So a screen is where you don't know what you're expecting. You set the experiment up as carefully as possible, and you then use whatever methodology you choose to try to understand what's different between the two conditions, and that can help you find something brand new that you didn't know before. So in this case, we took, after IV infusion, hyperglycemic mice or normal glycemic control mice with saline infusion. We isolated the islets and we labeled them and mixed them together, and then we identified, after a 2D gel, so this is very low throughput, we identified peptides of proteins that had changed in abundance in the hyperglycemic condition in the islets. And the proteomics was actually very carefully done, and was very high level of confidence for the proteins that were identified. And what you see here is from red to yellow that the proteins in the islet that were altered, and mostly these were increased by hyperglycemia, involved protein translations. These are proteins that are regulating translation of proteins, protein folding, protein degradation, and also peptide secretion. And so all of these, as you will immediately recognize, could be related to the folding of insulin and secretion of insulin. Overall, the signature from this red to yellow suggested activation of ER stress, endoplasmic reticulum stress. In particular, this stress response called the unfolded protein response. So the UPR, or the unfolded protein response, is basically, so this is my little diagram of the ER lumen, which there's this key chaperone called GRP78, glucose regulated protein 78, that helps proteins to fold. And it also binds to and keeps quiet these three transmembrane proteins, PERK, IRE1, and ATF6. And so during times of ER stress, not just in beta cells, but in essentially every cell type, when PERK gets activated, it's a kinase, leads to phosphorylation of eukaryotic initiation factor 2 alpha, EIF2 alpha, which actually suppresses new translation. So it decreases new proteins entering the ER. And then IRE1, when that becomes activated, develops RNA splicing activity, which is a very unusual thing to happen in this location in the cell. And its specific target is an RNA called XBP1. And so IRE1 activation leads to splicing of XBP1, which goes to the, then basically makes a transcription factor, which can turn on genes. And ATF6 is another unusual protein in that it's an ER resident transmembrane protein. When GRP78 is pulled away to fold proteins, ATF6 is released from the ER, and then it moves to the Golgi where it gets cleaved, and the cytoplasmic portion forms nuclear ATF6, which is then able to go to the nucleus and turn on and off genes. So if these three stress response pathways are, if their response is adequate, the end result is basically making additional rough endoplasmic reticulum to increase the cell's ability to make proteins in the secretory pathway, such as insulin. And then the stress will resolve. But if the stress response is inadequate, then this cell death effector called CHOP is activated and cells die. So the UPR is a very fundamental, highly conserved across species, from low to high, phyla, and also it's true in essentially every cell type. It's essential for cell survival and cellular homeostasis. And so its overall goal is to sense protein flux through the secretory pathway through the ER and to adapt the ER capacity to meet demand. It's very, very sensitive to small changes in ER protein folding load or also to redox status. And so the general paradigm in cell biology is this. So a stress to the ER leads to the UPR, which is adaptive, to hopefully maintain homeostasis. So there was actually already a lot of literature on ER stress in beta cells, and the literature pretty much is summarized by this: ER stress killing beta cells. And so there are a lot of, in vitro studies using high dose chemical toxins, also gene knockout studies in live mice, which are, as you can imagine, totally deleting a gene can be a pretty severe insult. And under most of those cases, when ER stress increased, beta cells die. And I'd also like to point out that making insulin is very energy intensive, and pro-insulin under stimulated conditions can represent up to 50% of the ER load in beta cells. And under high blood sugar, it's totally not surprising that this could get overwhelmed. So the first thing we did was we went back to our glucose infusion model. We confirmed that ER stress was activated. So all of these red panels here show that all three of the pathways molecularly were activated during hyperglycemia in vivo. But what we didn't find is increase in CHOP or dilation of the ER when we looked by electron microscopy. So this suggests that our glucose infusion model was causing adaptive UPR, but not decompensated ER stress and dying cells. And I didn't mention, but we really don't detect any increase in beta cell death in our mouse glucose infusion model. It's pretty much undetectable. So suggesting this is we actually have a window into the adaptive response in the beta cell, which is kind of new. So then we developed this hypothesis that maybe the unfolded protein response, so this response to ER stress, could be a sensor that detects insulin demand in the body in order to trigger beta cell proliferation when existing beta cells are becoming overloaded. This, I think, was controversial at the time and remains controversial, you should know. But I'll show you the evidence that we have so far. So the first experiment that we did, one of the first, was this. So these are Western blots. So looking at proteins from mashed up islets. And we have grown those dispersed islets in a variety of different conditions. So either low glucose or high glucose, and then with two different drugs in this case, thapsigargan or tunicamycin. Both of these cause ER stress. The high doses here are the usual doses people use. But we thought that probably to look at this adaptive ER stress, we should look at lower dose. And so we did a whole range of doses, including doses that basically caused very little observable changes to the cells. And what you see is that if you focus on the PCNA, this is proliferating cell nuclear antigen. So this is a marker of proliferating beta cells. When we grew the cells in high glucose, there was more PCNA. And in the lowest dose of thapsigargan, lowest 2 doses, you could see that the PCNA, the amount of proliferation antigen increased. But when we approached to the high dose of thapsigargan or tunicamycin, the PCNA dropped off. Then this CHOP cell death-like protein starts to increase. Even activated caspase-3 is increased. This is an actual effector of cell death. And then we even lose actin, and the cells are clearly not happy in the usual dose of thapsigargan or tunicamycin. Interestingly enough, when we repeated the experiment using the proliferative doses of thapsigargan or tunicamycin, the effect is glucose dependent. So if you culture and all of this is mouse beta cells, mouse islet cells. If you culture mouse islet cells in essentially normal blood glucose equivalent, you don't see proliferation when you add stress. But when you culture them in hyperglycemic conditions, adding subtle additional stress increases the proliferation percent, both for the thapsigargan and tunicamycin. And the converse was also true. So we used these chemical chaperones, TUDCA or PBA. So these relieve ER stress. And we had shown, I didn't show the data here, but when you culture them in high glucose relative to normal glucose, ER stress markers increase. When we culture with these two chemical chaperones, we those stress is relieved, and also the proliferation goes away. So we believe that the stress is actually a part of this, the trigger under these conditions for beta cells to replicate. We did a bunch of in vivo studies. I'm not going to share with you that shore up the hypothesis, but this is the main bones of it. Molecular studies, we've tried to identify whether PERK, IRE1, or ATF6 are responsible for the proliferative response to mild ER stress. And none of our studies suggest that PERK is responsible. But if we inhibit IRE1 or ATF6, we lose the proliferative response. And IRE1 is a special case because we can inhibit IRE1 and we lose proliferation. But if we overexpress IRE1 or we overexpress the spliced, pre-spliced XBP, we also lose proliferation. So this pathway seems to be, it's sort of a Goldilocks type thing. It has to be just right in order to get the proliferative response. But the ATF6 branch of the pathway, if we either inhibit it with a chemical or we overexpress it genetically, we gain proliferation. And if we knock it down, we lose proliferation. Wait, did I just say that wrong? If we inhibit it chemically or we knock it down, we lose proliferation. And if we overexpress it, we gain proliferation. So suggesting that nuclear ATF6 is a positive regulator of beta cell proliferation under stress conditions. Interestingly, we've also recently found that these two pathways are very tightly connected in beta cells. And so the XBP signaling actually requires ATF6 to turn on the XBP dependent genes. Okay, so just to summarize our ongoing project and future directions, but so our overall concept is this, that stress, as with everything else in the beta cell, is a continuum. We totally are in agreement with everybody that if you're in the red zone, where insulin secretory capacity is nowhere near meeting demand, severe hyperglycemia, beta cell dysfunction is happening, meaning they can't properly make and secrete insulin. Dedifferentiation happens as well. I don't have time to show you that. Beta cell death is high, beta cell proliferation is low. So this is, I think, the range that most of the prior literature on ER stress and beta cells resides. I also believe that this blue zone down here, where there's no load and no stress on beta cells, is perhaps not particularly healthy either. There's really no trophic input for beta cells to continue to work on making more insulin, secreting insulin, and to maintain a healthy beta cell mass. And so our work, I think, suggests that there is this green zone of moderate adaptive level stress that where the beta cells are engaged with respect to insulin production and insulin secretion, and that can also trigger an adaptive proliferation response without yet increasing cell death. So we have a number of genetic studies ongoing that some of them have been going on for a very long time, and we still don't have real data yet. So hopefully if you invite me back in a couple of years, I'll have more information to share with you. But several of the things we're focusing on for the future, first of all, is how does ATF6 alpha push mature beta cells to enter the cell cycle? And we're looking at molecular mechanisms of that. Second, so GRP78, I mentioned that as this key chaperone that helps, it actually is known to help fold insulin itself, proinsulin itself, but it also folds many other, it's a really critical chaperone in the ER across all cell types. So we have deleted that from beta cells using the insulin 1 Cree, so only in beta cells, and those mice do not do well. So the pups already are hyperglycemic by 2 weeks of age before they're even on, mother's milk in mice is quite fatty and low in carbohydrate, but they already have diabetes even before they wean to a chow diet. And that turns out to be, this is a severe ER stress, and the beta cells are dying, and that is dependent on a protein called junk. Hopefully that will be published soon. You'll notice that I referred to this as ATF6 alpha, where I hadn't before. And that's because we've deleted ATF6 alpha from the mouse and in beta cells. And the phenotype, and we've done this experiment now too many times for me to share. And sometimes there's subtle phenotype, sometimes there's no phenotype. So overall, if there's any impact of chronic deletion of ATF6 alpha, it's pretty minor. And so that's led us to wonder whether ATF6 beta, which you guessed it, is similar to ATF6 alpha. It's on a totally different gene, whether that might be compensating. So we have now generated a new mouse model that has LOXP sites around ATF6 beta. And that, so now we are testing whether ATF6 beta itself plays a key role in beta cells. And that, so far it's looking like it doesn't, that we don't think that ATF6 beta all by itself will be critical, because if you delete it from the whole animal, the mice are pretty much fine. But the point of making these mice is so that we can delete both of them. So we are in the process of breeding mice to do that experiment. And then there's one final mouse that we have made that I'm personally also very excited about, which so that nuclear ATF6 alpha, if we overexpress that in beta cells, we get a very, very rapid transcriptional response and also a proliferative response over the next few days. And so we've made a mouse where we can actually increase ATF6 alpha, this nuclear ATF6 alpha transcription factor at will in whichever cell type we want. And so we are just now beginning to study what happens if we turn on ATF6 alpha in beta cells in the mouse pancreas. So just an array of mouse genetic studies that will hopefully help us understand some of how ER stress and the stress response is regulated in beta cells, and how whether that can be used as a tool to increase beta cell mass. So I'll finish with just a couple of slides. Sort of perspective on what I've shown you. So I think this may reflect a new role for this very established, well-established, well-studied cellular mechanism of the unfolded protein response. So in cell biology, when people think about this, they always think about what's happening in a cell. So you have a cell and then you load the endoplasmic reticulum, the UPR is activated, and the goal or the result of that is increasing the amount of ER. So now this cell can handle more secretory pathway protein folding and load. So I think what our studies suggest is that at least in the beta cell, when you load the ER, we actually haven't been able to quantify, well, we haven't tried, but whether ER volume increases within any given beta cell. But what we do see is that it can increase the number of beta cells. So kind of expands the concept of the UPR from expanding ER capacity on a per cell basis to basically regulating cell number or tissue size. So I personally think this is a very elegant way that the body is tying a functional role of insulin production to the functional need of insulin demand in the body. Over the longer term, there's certainly a lot of gaps before any of this could be useful for people. I would love to be able to measure beta cell stress level in people using a blood test or radiology. A lot of people are working on radiology. I'm not working on that at all. I'm actually not working on the blood test actively either, but that's something we've thought about, and other people in the field as well. We would love to develop therapeutic tools that can modulate stress. Once we can measure the stress level, if stress is low, then maybe we would need to mildly increase stress to induce beta cells to replicate. If stress is high, then we may need to decrease stress to save the beta cells from dying. And also the tools could potentially be used to predict stress-related diabetes risk to allow preventative intervention. For example, you could imagine telling your patient, your beta cells are stressed, you had better stop drinking soda or whatever. Okay, in the last 2 slides, I think. So here we are in Weill, Cornell. This is actually all pictures of the Belfer building. Well, this is not. So my research lab and the Weill Center for Metabolic Health is on the 6th floor of the Belfer building. And actually, we're just about 1 week away from putting up our first job posting. If there are any scientists in the audience that, well, I certainly would never try to recruit anyone from my neighboring institution. But if you know of any beta cell biologists, or actually, we're not looking for beta cells, we're looking for anyone with an interest in metabolism and stellar scientists, we will be looking very soon. And we also, during the pandemic, made a wonderful move. I don't know if any of you had probably never visited the 80th Street office where Weill Cornell Endocrinology was housed previously. It wasn't on campus, it was far away. It was sort of like a private practice, but we've managed to move the division to Weill Greenberg, which is right in the middle of campus on, across from the main hospital building. I'm very excited about that. And also the Comprehensive Weight Control Center, I'd like to thank them because they are helping us move forward with clinical research as well in the division. And of course, all of our faculty fellows, diabetes education and staff. And then all of my colleagues and mentors at Weill Cornell in the lab. Well, Rohit Sharma now has his own lab. He is responsible for a lot of the ER stress projects going on in the lab in the past, and also we are working very closely together now and in the future. These are the current lab members moving some of these projects ahead. I moved from the UMass Medical School about a year ago, a little more than a year ago. These are my colleagues there. And I, again, would like to give one more shout out to Andy for getting me launched in my career. Thank you. And thank you all for listening. I'd be happy to take questions.
Thank you so much for a great talk. I think people can either unmute themselves because the default is mute, or type into the chat box.
So maybe I'll lead off if I may. So Laura, that was a great talk, and your pictures of the Cornell labs remind me exactly of the Adrian building too, only about 70 years newer. We're a little jealous. It's beautiful. So congratulations on that. So I haven't thought deeply about ATF4 and ATF6, but they both seem pretty interesting to me for a couple of reasons. So, I mean, ATF4 because it's downstream of PKA, and we have our own RNA-seq data. It's highly expressed in beta cells. And so we think there's this GLP-1 extendin-4 harmine story, ATF4 likely is downstream of that in some way. But ATF6, I really haven't thought at all about. So I was interested to hear that it drives, or at least it appears to drive beta cell replication. So do we, I think you said you were going to do this, or maybe you've done it. Have you overexpressed ATF6 in beta cells, and does it really drive proliferation, or is that a model?
Sorry, I didn't put the data in there. So we have a couple of different tools for ATF6 alpha, be specific on that. So ATF6 alpha, we have an adenovirus that overexpresses full length ATF6 alpha, and then we also have a construct we're calling DATF6, and this is through a collaborator at Scripps in San Diego, Luke Weisman, and he made this really beautiful system that is a DHFR, which is a dihydrofolate reductase, which is a non-stable peptide that is fused to the nuclear ATF6 alpha. So such as when you add trimethoprim, DHFR is stabilized, and then ATF6 alpha nuclear domain turns on. And he had shown before that genes turn on very quickly in that system, like known ATF6 targets. And so that we have an adenovirus, overexpressing that, and it works really beautifully. So even within 30 minutes, some of the ATF6 alpha targets start to go on when you add trimethoprim to the cultures. So using that model, beta cell replication clearly increases when we activate ATF6 alpha. So it's sufficient to increase proliferation. The in vivo studies are turning out to be more complicated. The N is still too small to be confident at all, but so we took that DHFR ATF6 and we, using CRISPR, put that in the COL1A1 locus, which is just a locus available to any cell type. We've mated that to insulin Cre. So now we have a Cre, sorry, lock, stop, locks, and then the construct. So we make the construct in beta cells, and then we can treat the mice with trimethoprim. And if you do that continuously, they get diabetes, at least with N equals 1 male and 1 female. We're thinking that probably pulsing it may be beneficial, but continuous and this is a common theme that most of the UPR pathways, if you blast them for a really high level or longer duration, that's not good. It has to be part of the kind of physiology in order to see beneficial responses.
You express this nuclear targeted version, it does drive beta cell proliferation if you pick the right dose in mice, I assume.
So in vivo, those mice, we've just started analyzing them. So we just all we've done so far is blast it for 2 weeks, and that's not good.
Right. But I mean, I'm asking just if you take isolated mouse islets. And it does drive proliferation. Oh,
definitely. Yeah. If you take mouse islets and you treat them with the TMP, beta cell replication increases over the 72-hour time period that we normally.
Does that work in human islets as well?
It does. Yeah.
And do you have any idea what genes ATF6 is, or ATF6 alpha or beta, whichever one it is? Has somebody done CHIP on that to see what the targets are? Or
okay, so CHIP. So one of the reasons you don't study ATF6 alpha and no one else does is because the antibodies are just a 0. So no, there's no CHIP of ATF6 alpha from anybody. We have actually tagged this construct that we now have in the mice. So in theory, we could do that experiment. What we have done is an RNA-seq experiment using the virus construct in mouse islets in culture, and we looked at 6 hours and 24 hours. And yes, we have lots and lots and lots of genes. There is no, that's sort of, I don't know, a quarter of the way through trying to sort out what might be responsible for the proliferation.
Cool. That's very cool. I didn't know that story. So thanks for sharing that. Yeah. See if anybody else has questions. Also, I think you misquoted me. I care deeply about mouse diabetes. Don't miss it. I feel equally passionate about rat diabetes as well.
So this is Alice Levine. Laura, thank you for a beautiful talk. It's not really a question, it's more of a comment. I was very excited that you did a little bit of the history at the beginning, because just coincidentally, I didn't even put it together that it's the 100th year. A patient of mine had given me the Michael Bliss book about the discovery of insulin. And I mean, for anybody who is looking for something fun to do, it reads like an operatic page turner, very analogous to the double helix, with all the infighting and people looking for credit and sharing Nobel Prizes and screaming at each other. And but it's also very informative and really gets you into that era. So thank you, and then for bringing us up 100 years later. That was a really nice arc.
Thank you very much. I should put that book in there too. I haven't read it myself.
It is actually, I mean, so I have read it, and so my impression was that Banting and Best were heroes and they worked in an ideal world. And the truth is they didn't like each other, but at the end they didn't speak to each other. Banting was never accepted into the Canadian and Royal Society sphere of influence, and basically went into exile in Edmonton. And the boss, McLeod, who I'm sorry to say was a Scotsman with an ego, basically tried to take all the credit of work that he wasn't even supervising. He happened to be in England during the time all this stuff happened, but when it turned out to be something good, suddenly he wanted to be the guy taking the credit. And so the two underlings fought with each other, fought with him, and
then there was a fourth person, and then as with the DNA structure, it turns out that all four of them really did deserve some credit, and other people that came before them. So anyway, great talk. Let's not get off on the Bliss book, but I really enjoyed how you put it all together.
Thank you. I tried not to put too much in there that you guys already all know. I did medicine grand rounds recently here, and that was, I had a bunch of stuff in there like, I don't think I should present this to a group of endocrinologists.
I don't know. I was thinking about it as you were speaking. I was thinking about generationally. I think in our generation, in my generation, Alice and I and Andrea probably know parts of that story pretty well. But I mean, we're old enough to remember when actually that stuff was still happening, you know? I mean, I remember people using acid test tablets to detect acetone. I remember people getting a single glass syringe with a needle and a bottle of insulin. So like, you know, good luck with that. Have a good life. I mean, so it is amazing what's happened.
So my mother's mother was an adult onset type 1, and she had she boiled her syringe, and I think 2 out of her 3 daughters became nurses, and that was a big influence on our family, I think.
Yeah.
Yeah.
I don't know, Nurali, back to you.
I think we're kind of up on our hour. Jack, I think, had just a comment that the Bliss book is very much worth reading, seconding what Alice and Andy were saying. But thank you so much for a wonderful talk. Hopefully we'll see you physically at Sinai, maybe once we're on the other side of this.
Excellent. And wait, hi, Mark. I just saw Mark Sperling pop up for a second. Where did he come from? Hi.
He's in New York. He's retired. He's in New York. He's affiliated with the PEDS department here. And then also Josh Tenenbaum has an interesting.
Hold on, we should unmute him.
Oh, you're muted.
I'll ask you one question, if I may.
Oh, yes, of course.
And that is the long survival of that second patient with supposed type 1 diabetes. It's really remarkable, given the impurity of the materials that were being used at the time, that she lived to 74. Are we sure that she had type 1 diabetes?
Well, she was what, 65 pounds, or was it 45 pounds at the age of 15 or something?
That doesn't mean that she didn't have, for example, a monogenic form of diabetes, which is real diabetes. She could have had a MODY3, or she could have had something else that gave her a pretty severe diabetes, but it really wasn't the destructive kind of type 1 diabetes. I just find it difficult to accept that she lived for 60 years with type 1
Mark, you're attacking the myth here.
I know, I'm just, well, we have lots of myths. And then we discover that there is a rational answer for the mythology. And I'm just looking for a rational answer, or at least asking a question.
I don't know. I certainly don't know the answer. But I do know that, I mean, there certainly are all, the Joslin medalists, and you're right that the tools weren't too good in the beginning, but I mean, I have a patient who's in her 70s now, and she describes that, how amazing it was when she could, check her blood sugar as opposed to just her urine. And she's doing remarkably well. So I guess, I mean, maybe this illustrates the spectrum of type 1 diabetes that we're all familiar with too.
Hey, Nurali, you should read Josh Tenenbaum's comment aloud. Actually, his mic is not working.
I don't think
I really can't see the comment.
Yeah, that was only to you.
I think it was a personal comment to you.
I see.
But Rhoda Coben has said that Joslin 50-year medalists are actually pretty common. I had lots of patients with clearly type 1 insulin deficient patients who died in their 70s and 80s. Great. Well, I actually have a patient scheduled in the next building at 9:40, so I should run. But thanks again for the invitation. All right. Bye.